So we will now discuss about the one more thing about the gene regulation in eukaryotes, and that are enhancers and repressors and the tissue specific gene expression. So enhancers and repressors, they both regulate and control the expression of specific genes that you have already have learned during the exon shuffling chapter. So enhancers are the DNA elements, they are the sequence, right, that facilitate or enhance the gene expression. Remember, these enhancers are the DNA elements. Both enhancer and silencers, they are the DNA elements that will facilitate. Enhancer facilitate to enhance the gene expression, whereas silencers will able to repress the gene expression. So first we are talking about enhancers, which are the DNA sequence which facilitate or enhance the gene expression. Enhancer promotes the binding sites for specific proteins, and those specific proteins are activators, right? Enhancer promote binding sites for specific proteins that will regulate the transcription process. These enhancers facilitate binding of the transcription complex to the promoter region. Enhancer normally facilitate, it allows or enhances binding of the transcription complex, the whole transcription machinery, to the promoter region. So this is about the enhancer. It mainly allows the transcription of that particular segment of gene or genes. Each enhancer is made up of short DNA sequence known as distal control element. Each enhancer is made up of short DNA sequences, and they are known as distal control elements. Activator normally binds to these distal control elements. Activators normally bound to these distal control elements and interact with the mediator proteins and transcription factors. Remember, during transcription I have talked to you about the mediator proteins. Mediator proteins interact with the RNA polymerase tail in eukaryotes, and also it this mediator interact with the activator. Mediator is actually a complex made up of at least 25 different types of proteins, and this mediator control the initiation process by regulating the kinase activity of TF2H. As I have mentioned during transcription, mediator controls the initiation process by regulating the kinase activity of TF2H. So these activators, they bound to these distal control elements. What are distal control elements? Distal control elements are those elements that make up your enhancer region. So within these distal control elements of enhancer region, activator bound to these distal control elements, and then activator interact with the mediator proteins and also indirectly with the transcription factors, right? We have also shown the diagram before, that how activators interact with these mediator proteins and transcription factors. Remember, during when we are talking about the transcription in eukaryotes, we have shown to you, I have shown to you about the mediator complex, and I have also shown that how activators interact with the mediators, and indirectly it interacts with these transcription factors for allowing the transcription process to proceed. Now, two different genes may have the same promoter but different distal control elements, thus enabling differential gene expression. So what the difference? If there is distal control elements, in that distal control elements activator would bind and would initiate the gene expression, and would initiate the expression of the genes by transcribing, by allowing the transcription factors to bind and carry out the transcription process. But this does not occur. If suppose there are two different genes, they have the same promoter, they have the enhancer region, but that enhancer region are different from one another, okay, because they have different distal control elements, thereby enabling differential gene expression. So again I repeat, all of the genes have a promoter region which are same from one another, but these genes also have the enhancer region, right? And each enhancer is made up of these distal control elements where your activators would bind and promotes the transcription process to proceed. But these distal control elements are different from one another, meaning the distal control elements of one gene, the distal control elements of one gene is different from the distal control elements of another gene, thus enabling differential gene expression. So in this way it interacts with the activators interact with these enhancer regions on the distal control elements and thereby allowing the expression of certain genes, right? Now let us talk about, okay, so enhancer are also the binding sites of your transcription factors. Enhancer are also the binding sites for your transcription factor. When a DNA bending protein, there is a DNA bending protein which will bend the DNA. When a DNA bending protein binds, the shape of the DNA changes. This shape will allows for the interaction of the activators that bound to the enhancers with the transcription factors, with the mediators, as well as RNA polymerase. So what happened? How does this activator interact with the transcription factor? That is due to the help of a DNA bending protein. So you can see in the figure, I will show you to you. Look at this figure. So in this figure you can see that a DNA bending protein here. See the DNA bending protein. So the DNA bending protein binds and bends the DNA so that your this enhancer region as shown here, the DCE are the short form for distal control elements, okay, distal control elements. Within these distal control elements, the distal control elements actually together form that enhancer region. Within these distal control elements, the activators have been bind as shown here, and it interacts with the what? It interacts with the mediator complex as shown here, okay, and then indirectly that these activators also interact with the transcription machinery. Here I have shown you the transcription machinery in this orange color, okay. Various transcription factor TF2A, TF2D, okay, TBP, which binds to a Tata box at the major group, right, minor group, okay. Then it also interacts with the other proteins also and allows the expression of the genes by promoting the transcription, okay. So this was about the enhancers mainly, okay. So remember, in these enhancer regions the activator binds and specifically it allows expression of the genes. What happen in case of repressor? Now just opposite of enhancer. Like prokaryotic cells, eukaryotic cells also have mechanisms to prevent transcription. Prokaryotic cells have simple mechanism, as I have shown you in the TRP operon, in the lac operon. Eukaryotic cells also have a mechanism to prevent transcription. Now there are some proteins known as transcriptional repressors. This repressor protein bind to the promoter region, it can bind to the enhancer region and block the transcription process. So like prokaryotic cells, eukaryotic cells also have mechanisms to prevent the transcription process. Transcriptional repressors bind to the promoter or it can bind to the enhancer region to prevent or to block the transcription process. Transcription process can also be blocked, as I have said in the previous video, by methylation, right? As I have demonstrated about the chromosome number 11 having two genes, one gene is silenced due to the methylation in their promoter region, right? So here the repressors bind to the enhancer regions and blocking the transcription process. Like the transcription activators, repressor respond to external stimuli to prevent the binding of activating transcription factors. So according to the external stimuli, it refers to the external signals conveyed by some signaling molecules conveying the message that we don't require this gene to be expressed now. At that time this repressor work, it respond to that message information carried by the some secondary messengers, right? And then it binds to this it binds to the activator. It's not activator proteins, it binds to the enhancer region or to the promoter region to suppress the transcription. Now they have also a site. Repressor also have a binding site that you have already learned it. I can show you that site. Okay, this is the picture that I have given to you during RNA processing, that is during your RNA shuffling, during your alternative splicing chapter. So here it has been shown that in the A figure you can see the green one is your mRNA and it shows the splicing site with repressor site. Splicing site with repressor site. In this repressor site, repressor protein can bind and can stop that splicing process, can stop that post-transcriptional modification process. Here it has not been, here it not shows the transcription process to be stopped, but RNA processing is stopped. These is also the same level of gene regulation in eukaryotes. So but the repressor does not bind. The splicing machinery, which has been shown in this figure, it binds and carries out the splicing process, right? The light shaded green one is actually your intron, which has to be removed out in order to get a mature mRNA. In the next, the same A figure, if you see in the left, in the right, you can see that the mRNA, their repressor site has been bounded by repressor, and repressor is preventing the splicing machinery to proceed its splicing process, and thus that RNA become unspliced. Okay, so here the expression will be occurring, but in this way actually repressor work, it prevents the transcription process at transcription level, right? In the next level you can see there is an enhancer, splicing enhancer. Okay, this figure is about respect to splicing process, whether it will occur or not. whether the splicing will occur or not in that particular intron exon junction. So here it has shown that there is a splicing enhancer region. There is also a splicing site. In the splicing site, the splicing machinery could bind. Obviously, it will bind the 5' splice site, the 3' splice site, the pyrimidine tract, polypyrimidine tract that I have said mentioned to you. Within the splicing enhancer, activators can bind and allows the splicing machinery to proceed the splicing process. Without binding of activator, splicing will not occur. So just in the same way, here I have shown about the splicing. In the same way, the repressor works in that same manner to prevent the transcription process also by binding to the enhancer region, right, or binding to the promoter region, or it can bind to its own site called repressor binding site called silencers. Okay, now it has been shown that in what way these eukaryotic repressors work. In what way the eukaryotic repressors work? Now this picture is a little bit of I have taken that image, but still you can able to see it. In the end I will give you the notes PDF from this lecture. So ways in which eukaryotic repressors work. Now transcription of eukaryotic genes can be repressed in various ways, and these include 4 mechanisms. First, how repressor repress the certain genes. First way, by binding of a repressor to a site on DNA that overlaps the binding site of an activator. By binding of a repressor to a site on DNA that will overlap the binding site of an activator. The repressor can then inhibit the binding of activator to a gene. Suppose repressor bind to a site on a DNA. That region, it overlaps the binding site of an activator. So as repressor bind to such site of DNA which has been overlapped with an activator, repressor inhibit the binding of activator to the gene and thus block the activation of the gene. So repressor here binding to such site in DNA which has been overlapped with the binding site of activator, that is your enhancer region. It has been overlapped to the enhancer region, and at that region the repressor has bind, so it will prevent the binding of activator to the gene, to that site, thus blocking the activation of that gene. In a variation of this theme, a repressor can be a derivative of the same protein as the activator. Now there may be a variation that repressor may be derived from the same protein as activator has been derived from that protein, but it lacks that activating mechanism. Right, I have shown to you during the splicing process, during the regulation, during the alternative splicing, right, that repressor have the same functional domains, the RNA binding domain, but it has not that domain to activate that splicing machinery in that chapter. Here also the same thing, repressor may be derived from the same protein as activator has derived, but it lacks that functional domain that will allow the activation of the gene. In another variation, an activator can bind to the DNA as a dimer. In another variation, an activator that can bind to the DNA as dimer can be inhibited from doing so by a protein derivative that retains the region of protein required for the dimerization but lacks the DNA binding domain. Such derivative forms the inactive heterodimers with the activator. Second is simple thing. Okay, what type of simple thing? It is saying that suppose to activate a gene, the activators have to be in the form of dimer. They should bind side by side to that enhancer region and form a dimer, but they are inhibited from doing so. They are inhibited from getting a homodimer by the repressors. So here you can see the figure. The first figure has been shows a competition between the activator and repressor. There is a binding site for the activator called activator binding site. Actually it is called DSE, distal control element, right? Or you can say the enhancer in a broad way speaking. There is a repressor binding site also. Repressor binding sites are actually silencers. Okay, so according to the first figure I have mentioned to you that repressor is binding to a site on DNA that overlaps the binding site of an activator. So what happened here, you can see the site where repressor should bind, it has been overlapped with the activator binding site. See here, you can see it clearly, it has been overlapped. Okay, now due to overlapping, the repressor binds and prevent the binding of the activator. The same thing can also occur in case of activator may binds and prevent the binding of the repressors. It may occur like that. So here we are focusing on repressors. So repressor binding to the site where it has been overlapped with the activator binding site, or DSE, distal control element, and thus preventing the transcription of that genes. Second is for activation of a certain genes, the activator would form a dimer that binds to a certain site on the DNA and allows the expression of the genes. But here what happened, dimerization does not occur. Here dimerization occur between the activator and the repressor. Why? Because they have been derived from the same protein, from the same genes, from the same protein. This activator and repressor have been derived, thus they interact with each other. The only thing that repressor lacks, if the repressor derived from that same protein where activator has been derived from, repressor here normally lacks that functional domain which will allow the activation of the genes. It lacks that functional domain. So interaction between the activator and repressor causes inhibition of the function of the activator, because the repressor is binding to a site of DNA besides an activator and interacts with the activator and occluding its activating region, means it is occupying with the activating region and prevents the activation. Okay, so this is a second mechanism. First is competition mechanism. As we can see very clearly that due to overlapping regions between the repressor binding site with the activator binding site, it is preventing the activator to bind in that site. Second is to formation for the activation of a certain gene, for the allowing transcription of a gene, the activator should form a dimer. Okay, as has been shown here, activator that binds to the DNA as a dimer can be inhibited. Activator that binds to the DNA as a dimer can be inhibited from doing so by a protein derivative that is acting as a repressor, that retains that region, that occupies that region, and the protein required for dimerization, but they lacks the DNA binding domain. So here the repressor is binding with the activator, but as they derive from the same protein, but they lack the DNA binding domain. Okay, such derivative forms inactive heterodimers with the activator. Okay, so these two has been now demonstrated. The third one is a repressor may bind to a site in the upstream of a gene, and by interacting with the transcription machinery at the promoter in some way that will inhibit the transcription inhibition. So a repressor can bind to a site upstream of a gene, and by interacting with the transcription machinery at the promoter in some specific way, that will inhibit the transcription initiation. So here repressor, just like activator bind to the enhancer region and interacts with the transcription machinery, repressor can also bind to the silencer region and can interact with the transcription machinery to prevent the transcription of the gene, to prevent the initiation also. Okay, let us see how it is saying. See, the figure is almost like same that I have shown to you during the case of activators. So see here, we can see that DNA bending protein, it bends the DNA, and there are some regions called silencer, as you can see in the top. Within this silencer region, your repressor is binding as denoted by R. It is interacting with the mediator complex and the components of the transcription machinery to silence that particular to silence, or you can say to inhibit the transcription process of that particular gene. So it is a type of direct repression. Okay, it is a type of direct repression. Okay, so this is the third mechanism that repressor may work. The third mechanism is a direct repression. It is not interacting with the activators. Okay, neither it is competing for binding with the site where activators bind, that has been overlapped with each other. Here in the third case, directly speaking, it is binding to the silencer region, interacting with the mediator complex, interacting with the transcription machinery to prevent the transcription of that gene. Okay, the fourth way is little bit from the first that I have talked to you during the eukaryotic gene regulation, the histone modification that can be done by the repressor. Repressor can recruit histone modifiers. Repressors can recruit the histone modifiers that can alter the nucleosomes in a way that can inhibit the transcription. Even they will inhibit the replication process also. So repressor can recruit histone modifiers. What are the histone modifier? Histone acetyl transferase, histone deacetylase, histone methylase, right? So the repressor can recruit histone modifiers that will alter the nucleosomes in a way that will inhibit the transcription, like by histone for example, repressor can recruit histone deacetylase. This repressor can recruit the histone methylase that can further cause inactivation of the genes by binding by removing the acetyl group from that particular lysine residue, as I have said, right? The histone get modified, and the DNA get tightly wrapped in to form a heterochromatin, right? So repressor can indirectly work here also. It recruits the histone modifiers, so that histone modifiers can alter the nucleosome structure that will inhibit the transcription. And for inhibiting the transcription, the DNA has to be tightly packed, coiled to the histone octamer to form the heterochromatin, right? So this work can be done by repressors. So in four ways repressor can work. First way I have shown to you. The first way is by competitive action. Okay, where your repressor is where the repressor binding site or the silencer region has been overlapped with the activator region, and the repressor is competing with the activator, so that who would bind first actually and carry out, and by binding it will prevent the transcription of a gene. Second mechanism, I have said that for allowing the expression of a gene, activator should bind to form a dimer and allows carry out the transcription of the gene. But here what happened, as the repressor has been derived from the same protein where activator has been derived from, it binds to form a dimer, to form a heterodimer, but it has one lacking in a functional domain, that it lacks that functional domain that will helps to activate the gene. So it is just binding with the activator to form dimer, but it will not carry out the activation of the gene to carry out the transcription. Third is direct repression. Repressor may bind to the upstream of a gene, which has been shown here. It directly bound to the upstream of the gene. Upstream means that is the promoter region, and after binding to the upstream it can directly interact with the mediator, with the transcription machinery, so that it will repress the transcription process. And where the repressor bind to the silencer region, obviously. And the fourth mechanism is indirect mechanism, where your repressor can recruit the histone modifiers, and histone modifiers will alter the nucleosome structure actually in a way that inhibit the transcription process. So this was about the second type of gene regulation in eukaryotes, by the activators and repressors.